Welcome to Hunter College, Harvey Lodish, founding member of the Whitehead Institute and professor of biology at the Massachusetts Institute of Technology, and my former mentor at MIT. Harvey's work has driven many novel applications in biotechnology and genomics. They address human health and diseases. He is the founder and scientific advisory board member of many biotech companies. Two of Harvey's trainees received Nobel Prize and the Lasker Award. And 8 are members of the National Academy of Sciences or Medicine. He's the lead author of the textbook Molecular Cell Biology, which has been translated into 13 languages, including Vietnamese. He's going to present 2 brief Gen talks, the first on the business of genomics and the second on Biotech and Genomics, New Therapies for Rare Diseases. Harvey?
Yeah, thanks, Robert. Let me not talk about Vietnam, although it's one of my favorite countries to visit and work with, but talk really about biotech and my involvement in biotech, but really the history of biotechnology and the many opportunities it's created for developing drugs for diseases that really have no cure. I was privileged to get involved in biotech very early in the 1970s. And in the second talk, I'll describe my experiences in putting Genzyme together, joined by 7 MIT faculty colleagues to develop drugs for rare diseases. And I'll talk about a few of my other companies that are also focusing on drugs for rare diseases and some of the issues about them. Also, I've been privileged to chair the research committee of the Boston Children's Board of Trustees. I see many of the gene therapy developments that are going on there that I will talk about a bit. And from my 10 years of work with the governor of Massachusetts on the Mass Life Sciences Initiative, I've had a lot of experience in how the state can best develop biotech and really create jobs as well as develop new drugs. So perhaps if you have questions on those, I could answer them. But let me begin with some of the fundamentals, because what many people don't understand is that universities and hospitals, such as this one or Children's Hospital, don't actually develop drugs. What we do is the basic science that clarifies a particular disease and generates what might be called a lead. That is, some substance, could be a protein, a small molecule, that treats the disease in a mouse or in a culture system. But to actually develop a therapeutic almost always requires a for-profit company, someone who can manufacture enough of the substance under GMP conditions to satisfy the FDA, someone who can run all the preclinical experiments, the safety experiments, the clinical trials, manufacture the material, and market it. So there is a clear transition between biotech and translational research in institutions such as yours and mine and companies. And I'm going to place great focus on faculty entrepreneurs. That is, faculty like myself who take discoveries out of our own laboratory or out of colleagues' laboratories and build companies. And some of what you need are shown here. Obviously, proprietary intellectual property. One needs a business plan. Many students who want to work in biotech don't quite understand that you've got to make money. You've got to convince people that not only are you going to make a drug, but you have a budget. What's a budget? And you have an idea of how you're going to sell it so that somebody's going to buy it and pay all your costs. Because I hate to say it, companies exist only to make money. If you're not going to make money, nobody's going to fund you. Sorry about that. You need other things, including venture capital, to provide the money. We can talk about what governments can do, which is actually surprisingly little. And a key, and I was glad to hear you have it here, is the fact that I, as well as my colleagues at MIT and many other universities, have 1 day a week where we can consult. I do a lot of my outside activities at not-for-profits, such as Boston Children's, but I probably spend 2 days a month, maybe a little more occasionally, on my several startup companies that I'm currently working with. And here's really the beginning of the underlying science. That is, biotechnology grew up in the 70s through recombinant DNA technology. Before this revolution in science, all of the drugs were small molecules made by techniques of organic chemistry, essentially pills that you eat. And what a number of us realized as recombinant DNA, just the ability to clone and express recombinant proteins, was being developed in our laboratories, that this could be the basis of an industry, or at least of a number of therapeutics. And I'm going to go into that, and also the parallel discovery in the 70s of monoclonal antibodies. When Cesar Milstein discovered it, and Cesar was a good friend until he passed away, he didn't understand, he didn't see that these could be used as therapeutics. He never patented it. Many others did. And this has led not just to a huge industry, but to a number of novel therapies. And of course, with the sequencing of the human genome 20 years ago, this opened up a huge new set of opportunities in which nucleic acids themselves, gene therapy, oligonucleotide therapies, could be done. And as I'll describe in the next talk, we're seeing the fruit of these now. Gene editing, and who knows what else is on the horizon. So let me focus on proteins as therapeutics, recombinant therapeutics, and monoclonal antibodies. These are today the big drugs of the biotech industry. Just to give you a bit of background, aspirin is a natural product. It exists in the bark of a certain tree, and it led to, well before recombinant DNA, to a number of drugs with similar modes of action, blocking prostaglandin synthesis, treating various inflammatory diseases. One of the consequences of the recombinant DNA revolution is not just proteins therapeutics, but proteins as targets for drug development. And I've listed here some of the many small molecule drugs that used as targets well-cloned proteins with well-known functions in disease. For instance, prostaglandin inhibitors for inflammation. If you didn't know it, the erectile dysfunction drugs were isolated as inhibitors of a cyclo-GMP phosphodiesterase. Actually for baldness, but that's another story. And again, almost all antibiotics now are developed with targeted proteins from bacteria. And of course, many receptor inhibitors and agonists are small molecule drugs using recombinant proteins. But it's really these companies that were started in the late 70s that developed these first biotech products. And the initial ones were all replacement proteins. That is, insulin as a replacement for individuals with diabetes who couldn't make sufficient insulin, replacing porcine insulin and other things. Clotting factors were originally made from pooled blood. And of course, there was a tragedy when some of the pooled blood had HIV. This was all taken care of with recombinant production of these. Erythropoietin, which I was involved with as a stimulus for red cell production used in kidney failure. I'll talk about glucocerebrosidase later. And these are all produced now, almost all of them, in cultured mammalian cells. We have to use mammalian cells because of the post-translational modifications such as glycosylation. Almost all of them use recombinant CHO cells grown in serum-free medium. And I'll talk in the next hour about the Genzyme Cerezyme drug that I was involved in developing. Now, I show this slide to point out that in the United States, 10 of the 15 largest selling drugs are recombinant proteins. And if you look at the top, Humira, a monoclonal antibody to TNF-alpha, sells for $20 billion a year. That is not a typographical mistake. That is $20,000 million of this one recombinant protein. And if you look further down the list, there are actually 3 inhibitors of tumor necrosis factor alpha that collectively sell for over $30 billion a year. Now, I'm not here to argue drug pricing. We can discuss that offline. But that is a hell of a lot of money. And yet, it's used because it treats some rather difficult to treat disorders. And since my experience is most of you have no idea what TNF-alpha does, it's a normal immune cell product. It's an inflammatory hormone. It induces inflammatory immune responses in response to wounds or other types of injuries, but it also is overexpressed in patients with diseases such as rheumatoid arthritis, other types of arthritis. It also plays a role in a number of autoimmune disorders. 2 of them are monoclonals. I don't like this mouse. I'll just mention Enbrel for a moment because it's a recombinant fusion protein that is based on a knowledge of the TNF-alpha receptors. Let me use this pointer. So basically, here's tumor necrosis factor alpha. Here's its receptor sitting in the surface of the cell. And in this case, 3 molecules of TNF-alpha, of receptor, bind 1 TNF-alpha. You can make the protein as a recombinant secreted protein, and it will bind TNF-alpha. But the protein cannot be used as a drug because it immediately disappears from the circulation. But if you modify the carboxyl terminus of the protein to put on the Fc or constant domain of a human immunoglobulin, you now have a chimeric protein that binds TNF-alpha and withstands, it lives in the circulation for many days. And as you see here, this is $35 billion, but a lot of patients treated, a lot of people walking around and functioning because their autoimmune disease or their arthritis can be treated by these drugs. And let me state, I have no interest in any of these companies. I'm just telling you this as facts. But you can see where this recombinant protein revolution has taken us. And the developments continue. There's a great deal of work modifying recombinant proteins to make them work better. As you saw, you can add carbohydrate chains. you can fuse the protein to an antibody with erythropoietin, a group at Amgen showed by adding additional carbohydrate chains, you can stabilize the hormone for almost 2 weeks. And that drug, darbepoetin, is in the clinic. You can add polyethylene glycol and stabilize many proteins. So this is an ongoing process of drug development that is based on the cloning of these recombinant proteins and monoclonal antibodies that began in the 70s. Let me just end with a bit more about biotech, because there's a lot of interest here and elsewhere in building biotech communities. And we have in Boston the largest conglomeration of biotech companies centered around Kendall Square. This is Kendall Square. It's the intersection of Main Street and Vassar Street. My office is right there. Okay? That is Kendall Square. That's the Whitehead Institute. Surrounding us is MIT and several hundred biotech companies, roughly 300 within a mile radius, plus 18 of the world's largest biopharmaceutical companies have offices or laboratories there. And there's a reason for it, because everything is clustered together. The next hour, I'll talk about Rubius, a company I recently started that is now public. But what I want to point out here is, here's my office at Whitehead. I started it with a group of venture capital people whose offices are a 5-minute walk down Main Street. And we started the company in an incubator supported by the Commonwealth of Massachusetts called Lab Central. I now have 2 other companies in Lab Central. That is, again, a 5-minute walk in the other direction. And I'll talk a bit about Lab Central. And when we got too big for Lab Central, we moved into rental space right there. And all around us is MIT, a number of venture capital firms, patent attorneys, and so forth. This is the same area 40 years ago. It was literally an industrial wasteland. It grew up organically by private investment driven by all of the small companies that my colleagues at MIT and I helped start. The Whitehead Institute we built we opened in 1983. We were the first building there. And a number of us at Whitehead have started companies. In fact, over half of my faculty colleagues at Whitehead have started publicly traded companies. So my involvement as an entrepreneur is by no means unique. Whitehead, in turn, hosted the Human Genome Project. Eric Lander was at Whitehead when the genome was cloned and sequenced. That gave birth, in turn, to the Broad Institute, the large genome center that is next door to Whitehead. And I just want to point out some of the individuals in this picture, because it points out what one needs to do to get together to form a biotech ecosystem. So if I go from left to right, that is Eric Lander. Next to Eric is Susan Hochfeld, who's the president of MIT. MIT and Harvard don't get along too well. We're competitors. On the other hand, in biotech, we work together. Here is Drew Faust, who was then the president of Harvard University. This is Eli and Edith Broad. Eli, the Los Angeles-based guy, who donated now $700 million to get the Broad off the ground. Next to her is Deval Patrick, the governor of Massachusetts. You see what it takes. And next to the governor is David Baltimore, the Nobel Prize winner, who founded the Whitehead Institute, president of Caltech, and chaired the board of the Broad Institute. So you really need to get all these groups together to build up this kind of ecosystem that you want. And one of the things that came out of the Mass Life Sciences Center, this board that I chaired, was Lab Central, the incubator where many, now somewhere around 50 companies have been started. There are many other incubators in the Cambridge-Boston area. Lab Central is particularly exciting because it has an open lab with about, well, now with the addition, probably 40 lab benches. This looks like any lab in any research building. But your company is right here. $6,000 a month, you rent a bench in Lab Central, and that's your company. It's fully supported. You have access to all the equipment that you would expect to find in a lab. They don't have animals, but all the heavy equipment's there, cold rooms and fax machines and DNA machines and microscopes. And you can have 1 or 2 people working at the lab bench. You start the company on Friday, and you're in the lab on Monday. If you get a little too big, you can move into a 500 square foot lab, close the door, and have 10 people. And if you get a little bigger than that, there's plenty of real estate around MIT that you can rent. So when Rubius, I'll talk about it in the next hour, got too big for Lab Central, we moved to a small lab here, then here in this building, a slightly larger lab 2 years later here. At this point, we had about 60 people. Once we had an IPO, we moved into a new building that was just built by Alexandria Real Estate over there. So what I hope you've seen from this is at least an overview of how what I hope you've seen is an overview of how biotech certainly began in Boston, and how it's grown over the past 40 years, and a glimpse of some of the treatments that have come out of it. And in the next talk, I'll discuss rare diseases and some of the specifics of Genzyme, Rubius, and also talk about some of the gene therapy treatments that are taking root and are being successful in Boston. So thank you, and I'm happy to take questions. How do we work this, Robert?
Just
ask questions.
Answer the questions.
Yes.
So the idea behind having the state-funded Lab Central incubator is the idea that it's going to bring an economic engine to the state?
Thank you for asking. The Mass Life Sciences Center was Deval Patrick, Governor Patrick's vision. I was privileged to chair the Scientific Advisory Board. We reported to the Secretary of Economic Development. In other words, it was to bring jobs into the Commonwealth of Massachusetts all over the state. And there, midway through 2013, there was an economic analysis done. And it was very favorable. It was done by the Boston Foundation. And one of the features of the Life Sciences Center was job creation by tax relief. So just to give you an idea of the number, companies would propose to hire certain numbers of people and in return get tax relief. And they did an economic analysis of that. I think at the time there were 9,000 or so new hires. And the bottom line was, for every dollar the state lost in taxes, if you looked at the taxes that the new hires would pay over a 5-year period, just state income tax, over a 5-year period, so just straight arithmetic, you get a return of 65 cents on the dollar. In other words, it worked. And needless to say, a lot of drugs were created and a lot of devices were created. So it was really pushed as economic development as it should be. Because the state is not in the business of developing drugs. Okay? Great question. Keep going. Yes?
So we are basically undergraduate teaching institution with
Yes.
graduate students. So I'm interested in your view of education in such an institution. Should it continue the conventional, learning the basics? Should we add new aspects?
I would argue both. I've been teaching at MIT 52 years, and I've always taught undergraduates by choice. So I think the basics are important, but to convey some of the opportunities that the science creates. And I find, as I suspect you do, that a lot of the students are bored by biochemistry and cell biology and genetics. They just don't see it. On the other hand, I'll show you in the Genzyme drug. That's a cell biology drug, you see. When I talk about Rubius and how we engineer red blood cells, that's a problem in hematopoiesis. The same is true with gene therapy. And certainly in the textbook that I write, we try to introduce these things. So I think the more connections you can make, that this basic material actually leads somewhere, is very important. And I certainly tried to do it when I taught cell biology, as I did for many years. I always tried to do it. And I think that's really the key in an undergraduate education. I'm sure you're shaking your head, or you have a question?
Well, I just wanted to make a comment.
Please.
In biology, we have a biotechnology track, and what that involves, I don't see the director of that program, but I hired her to develop a biotechnology program within the biology department for
Right.
undergraduates, and they spend a year after being undergraduates as master's students. And now what happens is there's a special biotechnology lab, and our students have to do an internship, and they're hired all the time.
This is wonderful. Now, this is thank you for mentioning. This is the second part, which is to train students for biotech. If I go to the Mass Life Sciences Center, this is the state one, I actually helped put it in place, an internship program where any student in Massachusetts, there's a website that sort of functions, I'm told, like a dating service. The students and the companies find each other, and then if they agree, the state pays them $7,000 a summer to work. And the condition is the company must have less than 100 people. So they're in a startup or mid-stage company, and many of them get hired, yes. I think this kind of pre-professional training is excellent, and I think what you'll find is that many of them will work in companies for a while and then go to graduate school. And I think that's wonderful. It's the way, they need to see the outside world. And just parenthetically, over the years, I'm often invited to lecture at universities or colleges and have lunch with students. And we always wind up talking about these things. And I asked them two questions. I said, how many of you have ever set foot in a company? And the answer is about 10%, usually a summer internship. And when the student talks about it, they rave. And then the second is a more difficult one. I said, how many of you, and these are usually graduate students, how many of you would feel comfortable going to your PI and asking for advice on careers outside of academia? And that's tough. Most say no. The vast majority everywhere say no. Okay?
Students are going here.
Maybe Hunter's different, I would hope, and it may have changed.
It's just the economy.
Yeah, wait, I see your hand up there. Yeah, go ahead.
I'm just curious about something that was mentioned in the beginning. I was wondering how you had a slide saying that for a startup company, you need a strong leadership in biopharmaceutical expertise and a scientific advisory board.
Yeah.
I was interested in the scientific advisory board. How do you find people that are morally capable and ethically conscious about the experiments you want to do?
Again, the scientific advisory board, the top ones are all academic scientists who are in the field. And that turns out to be key, because oftentimes the direction of the company may shift because of new opportunities and so forth. And the composition of the SAB often changes as the company develops. But my experience is that this becomes very important, and particularly at the beginning, where the SAB can meet almost weekly, if not, every couple of weeks, to go over the progress that the company's making. Because many of the workers in the company are young, and some adult supervision from experienced people can be helpful. As the company grows, the SAB becomes less important. Yes?
I have a good question of curiosity. Obviously, all of this economic growth is very important, all of that. But how do you feel that goes back to the origin of the basic science that was needed to get to this point? So it's like, how much of that trickles back into academic labs to help keep the line of scientists coming? Because there is this, I'm sure you're aware, all of this debate now, and I'm sure you didn't want to talk about the cost of drugs, but the idea is that if the original discoveries were powered with public money from NIH, shouldn't that somehow benefit them back to the universities where the knowledge required for that
I
was
mean,
generated?
the way it goes back to universities now is in licensing fees. And in other words, the original discovery in the university is the basis generally of a patent application. And the patent is licensed to the company which actually develops the drug. I mean, what you said is only partly true. The universities don't discover the drugs.
No, I know. I'm talking about the lab.
Yeah. And in the licensing agreements, they vary among institutions. A standard one is the university itself gets a third, the department or laboratory gets a third, and the inventors get a third. So it depends on what system each university has. There's no fixed protocol for this. Some are very different. And in some cases, it can be considerable. I mean, Children's, as an example, developed what became lenalidomide, which is one of the largest. It was on my list. And they received, it was actually a one-time payment of around $60 million, which certainly helps. It is, however, a small fraction of the revenue of lenalidomide over the lifetime of its patent. But that's what it was. But that's one of the blockbusters. So there's that provision. Other than that, there is no way, you see, other than philanthropy. And many of us who have stock and so forth have given some of it back. Robert?
So could you speak to how, if the state and the government invest in the development of these biotechnology companies?
No, they invest in the incubators.
Yes.
Not the companies.
Okay. That doesn't make any difference there
Sorry.
with respect to my question.
Okay.
What activities or what positions would you recommend that they take to ensure that the benefits redound to all people? For instance, as you know, with David Reich's experiments, he's been prohibited from having access to the Navajo people, their information.
Yeah.
And in many cases, African American DNA is now being used to study diseases and so on, which will likely benefit them, but in other cases that may not be the case.
Yeah, I have trouble, like many people, with that question. And I don't know how you monetize yet this type of DNA sequence. That's what it comes down to. It's clearly important, and clearly you have to be sympathetic to the groups. I mean, perhaps the best way I can answer it is the way Children's Hospital does genetic analysis of diseases in specific ethnic groups, where the team includes anthropologists and sociologists and works with the leadership. These would be consanguineous communities where there are genetic defects that lead to autism, neurodevelopment, and so forth. Very sensitive to it. But that's academic research. How you would deal with it if you're actually using that DNA sequence information to get a drug, I'd have to think about that. I think it's very complicated. I think the problem is people have not been culturally sensitive in general to the fact that DNA has a lot of information in it, and that it really is part of different groups. Please.
So you see this boom in companies, and it just reminds me of like all these other types of booms in small companies during the digital age. Like, how many of these incubators will be successful, and how much of is it just one patent, one idea, throw money from a venture capitalist like a line item, and then they fail and move on?
Excellent point you're raising. To give you a few numbers, with the Mass Life Sciences Center, we had a program of supporting very young companies with a one-time $1 million grant. To get them, they had to specifically describe what they would use the money for to get them over a hurdle so they could get larger venture funds. And if the company were successful and ultimately raised $21 million, they pay back the loan. Now, I describe that because for every cycle, we did it twice a year, we would get perhaps 30 applications. And we would wind up funding 4 or 5. And many of the companies never should have gotten started, bluntly. Either the science was mediocre or poor, or they had no idea of what it was to run a business, or both. So I had everything reviewed by VCs and other business types, academics who were running companies and so forth. And many of the companies that get started never grow. One of the rules of Lab Central has been very successful. The Mass Life Sciences Center was very successful because over half the companies have paid their loans back. But we're very careful in what we select. And the VCs are as well. It's trivial to start a company. You just need to file paperwork in Delaware or someplace. But to grow beyond 2 or 3 people is difficult. And I visited in many parts of the world that have 2 or 3 people, and they've had it for 5 years. That doesn't work. At Lab Central, you have 2 years. Either you die or you leave and grow. So you have to be careful. Not every good idea merits a company. Sorry. I'm tough. People come to me all the time with ideas, and I tell them MIT pays me a lot of money to tell them what's wrong. And I do. And sometimes they come back and they fix it. Most of the time they get angry with me. Okay? But that's the way it goes. You saw my track record. I put, I'm, 9 and 2. That's not bad. More questions? How much? I don't know. You're in charge, Robert.
We should switch to your other topic.
Can I get some water? Yeah. Okay.
Here's your water.
Okay.
Free?
Free? Okay. Okay. I'll use this. When I helped set up Genzyme just about 40 years ago with 7 colleagues at MIT, we developed the first drug for a rare disease, a drug called Cerezyme for Gaucher disease. And over the course of many years, I've helped start a number of biotech companies. And virtually all of them, at least at the beginning, had as their goal understanding and developing cures for rare diseases. And many of the successful small companies have drugs for diseases that are relatively rare. So to begin, a rare disease is not all that rare. The United States passed many years ago what is called the Orphan Drug Law, which gives marketing privileges, not patent privileges, but marketing exclusivity to companies who develop drugs for diseases that affect under 200,000 people a year. So that is, in a sense, the legal definition of a rare disease. But that's an awful lot of people, and that's an awful lot of disease. Roughly 10% of Americans are thought to have a rare disease. Many of you would know them. I'll show you a list of some, but they're not all that uncommon. Yet we have 6,800 that are known in the medical literature, and the fraction, about 10%, is common throughout the world. Now, we understand a great deal about a few rare diseases. I've listed some of them up here. Some are very well known, like cystic fibrosis, phenylketonuria, I just talked about, Duchenne muscular dystrophy, other muscular dystrophies. The point is that these diseases were studied, discovered, and cures developed for them for Caucasian populations. There has been to date no systematic study, hardly any studies at all, of rare diseases that affect non-Caucasian groups. I'm speaking of East Asia, South Asia, Africa, many isolated ethnic communities throughout the world. There is a huge biological opportunity, a huge opportunity to understand human biology by understanding these diseases, and particularly with the emerging technologies of cell and gene therapy to develop cures for many of them, and I'll go into it. I mean, for instance, Tay-Sachs, as I'll show you, I'm sorry, not Tay-Sachs, cystic fibrosis. I was with the president of the Chinese Organization for Rare Disorders in Boston, and I said, would you like to visit Vertex? They've developed remarkable drugs to cure cystic fibrosis. He says, no, there's no cystic fibrosis in China. Okay? And what makes rare diseases attractive, as it were, for biotech is almost all of them are genetic in origin, and almost all of them initially affect young people. And that means they're potentially treatable by some of these modern technologies. Now, I mentioned studies in Caucasians. I'll just show you a couple. Because of intermarriage, autosomal recessive diseases spread in isolated populations where consanguineous marriages are common, you can see countries like Iceland and Finland where people tend not to, at least historically, not to venture very far. diseases that are really found nowhere else. And one of the reasons for studying rare diseases, even if you can't find a cure, is by screening the affected population, one can often prevent the birth of children with these diseases. And I point out here one, it's one of the oldest ones, that was developed in the Orthodox Jewish community in Brooklyn, which is affected by diseases like Tay-Sachs and Gaucher and many others. And the point here, or a point here, is that it was organized by the religious authorities, the rabbis, who convinced their followers that this was in fact something that was important to do. And it is, at least as I am told, the rabbis who consult with couples who are planning to marry. And certainly in African American communities, screening for sickle cell disease certainly here is growing, and hopefully it will prevent occurrences of that disease. And finally, there are very few of these diseases that have treatments. And that's really a point that I want to go through, particularly now that we have technologies that can be used to treat these diseases, providing funding and other things are available, other resources are available. I'll start with a discussion about Genzyme, which is a company I started in the late 70s with 7 MIT faculty colleagues. And we were all pretty good scientists, but the real credit goes to this gentleman, Henry Termeer, a Dutch MBA, who saw that, or had the vision, that rare diseases not only could be treated with some of the emerging protein technologies, but that you could build a company on that basis. And just a bit about Gaucher, which is the first of these genetic diseases to be treated. It primarily affects descendants of Eastern European or Ashkenazic Jews. And it's an autosomal recessive disease lacking a lysosomal enzyme, glucocerebrosidase, which breaks a particular glycolipid, glucocerebroside. Depending on how much residual enzyme is left, you can have a severe form of the disease, types 2 or 3, that lead to brain damage or death. I'm telling you this for a reason, so just remember it. Or if there's a small amount of enzyme remaining, you have the peripheral disease, which is characterized by hepatomegaly, a large liver, a large spleen, weak bones. The bone marrow fills up with macrophages with glycolipids inside, and so forth. And it's a cell biology drug, and I love it. It's a recombinant protein made in CHO cells. But the idea is not just to have the protein, but to be able to inject it and have it be delivered to the macrophages that are the cells that need it. So as I'll describe in some detail, the carbohydrate chains are modified such that the residual carbohydrate chain with its mannose residues binds to a mannose receptor on macrophages, and the enzyme then is internalized, delivered to lysosomes, takes up residence, and reverses the disease. And here's the cell biology. Macrophages, like many body cells, have pattern receptors on their surface that recognize foreign molecules. In this case, sugars with mannose residues that are not normally found on human proteins. As it happens, fungi like yeasts have cell walls that are built of polymannose sugars. And many other pathogens have this type of cell wall. So the macrophages evolved to have a mannose receptor. You can see the macrophage on the right binding to a yeast cell through its mannose receptors and ingesting it. And what is shown on the left here is a typical oligosaccharide, an asparagine-linked oligosaccharide, attached to a typical recombinant protein, say glucocerebrosidase. And the genzyme drug, the Cerezyme, takes this recombinant protein, treats it with 3 enzymes that successively remove sialic acid, galactose, and acetylglucosamine, leaving this trimannose core that is a substrate for the macrophage mannose receptor. So therefore, this molecule, when injected, goes directly to macrophages, where it's transferred to lysosomes and reverses the disease. So this is one reason I'm telling you about the Genzyme story. There's another reason. A rare disease is rare unless it's in your own family. I have 7 grandchildren. One of them was diagnosed in utero as having Gaucher disease. That's Andrew. His mother and father, his mother, our daughter, parents are of Ashkenazic Jewish descent. They turn out both to be carriers. Our daughter freaks out, does not know her father knows anything about the disease, has amnio, and has me take the call from the genetic counselor who says the child your daughter is carrying has Gaucher. Well, it's a long story that's been written in several articles. I could send them to you. But basically, we could tell from the DNA that it was going to be the type 1 case, type 1 type of the disease that could be treated with Grandpa's drug. And Andrew is now 17. He just rode his bicycle with a group of high school students from South Carolina to Florida. He had to stop in Oklahoma City. His mother had to fly down with an infusion, and he had to go to Oklahoma Children's Hospital, but he's okay. So the point is, it does work, and you never know who's going to be helped by a drug you helped develop. And Genzyme was sold to Sanofi for $20 billion. That is $20,000 million. You guys know what $1 billion is? Okay, it's a lot of money. Anyway, but Genzyme makes a number of recombinant proteins that are used to treat other rare-ish diseases, all lysosome storage diseases. Briefly, let me go through one of my newest companies, which is Rubius, which is developing treatments for other genetic diseases based on genetically modified red blood cells, where a transfusion of a small number of red cells that have been genetically modified can be used to reverse a genetic disease. In fact, we think several genetic diseases. Had an IPO last summer. It's again worth somewhere around $1.5 billion. So I can be pretty confident that everyone in this room has been tested for phenylketonuria. It's done as a toe prick at birth, looking for elevated phenylalanine levels in the blood. It's an autosomal recessive disease missing an enzyme that catabolizes phenylalanine. The elevated levels of phenylalanine in the circulation can cause brain damage. There's no treatment. There's an unpalatable diet. I've tried it. That costs about $100,000 a year. And those of you who are adolescents or have raised adolescents know that they don't keep to a diet very well, particularly one like that. So it's a problem. And the idea for this project came out of work in my lab studying red cell development. And I don't want to go, or I could, but I won't go into the details. But for many studies, we needed a system where we could take human stem cells, bone marrow-derived stem cells, hematopoietic stem cells, put them in culture and have them divide, in this case, 14 or 15 times, and make normal red cells that lose their nucleus. So we needed this for our experimental work, and eventually came up with such a system. It was published a number of years ago in Nature as part of a very different project. But you can see the normal-looking red cells that were made in culture. And in the course of, excuse me, in the course of doing these experiments, we would use recombinant DNA techniques to add or delete genes from the red cells for experimental purposes. And then it occurred to me that one could actually use red cells to deliver enzymes or antibodies into the human body for long periods of time. Because red blood cells, new red cells, have a lifetime of 3 to 4 months. And they're lost their nucleus. The last stage in red cell development is the cells kick out their nucleus. So you can add whatever DNA molecules you want during the development of the red cells, introducing foreign genes, and have all the genetic material kicked out, the nuclear DNA in mitochondria. So there's no chance of some weird cell crawling out of the transfusion. And we consider them 7 micrometer diameter microparticles. They're very flexible. They have all the advantages that you tell your students about. And what we simply did, or Rubius did, we did many other things, but Rubius did this one, where they used standard recombinant DNA techniques to introduce into the red cells a potent bacterial gene, a gene for a potent bacterial enzyme that degrades phenylalanine, not the missing human enzyme, but a better one. And culture these stem cells in vitro. The resulting red cells lose their nucleus. They have normal circulatory lifetimes and so forth, but they have in them an enzyme that will lower serum phenylalanine. And in this sort of mock experiment, you incubate these red cells with serum spiked with phenylalanine, and it goes right down. So gives you an example of what you can do. Let me end with some discussions about gene therapy. I mentioned some time ago that I'm on the board at Boston Children's, and I'm privileged to watch close up a lot of their gene therapy trials. And I find it absolutely remarkable that with the right combination of basic lab scientists, clinicians, and companies that manufacture the viral vectors, you can treat kids with diseases like the bubble boy. This child, I think many of you know, was born with X-linked severe combined immune deficiency. It's a loss of a receptor chain. It's called the IL-2 common gamma chain. It's a component of the IL-2, IL-4, IL-13, and many other receptors. Without it, you have no functional B or T cells. And this child lived in a bubble until he was 7 years old and died. These kids are treated now. It's essentially a bone marrow transplant. It is a bone marrow transplant. It's an autologous bone marrow transplant where you take stem cells out of the patient and you infect them with a lentivirus that contains the missing gene. Now, a code word. Lentivirus is HIV. Don't panic. They use HIV because it's very good at infecting immune cells. But it's stripped of all the disease-causing material. On the other hand, you have to change the name. So it's called a lentivirus. But it has inside a promoter and the cDNA for X-linked SCID. And you simply infect the hematopoietic stem cells in culture. It infects enough of them to deliver into the nucleus the missing gene. And then the reconstituted stem cells are transplanted back into the patient. This is a routine procedure in a well-equipped hospital because it involves first taking after the stem cells are withdrawn from the patient, you have to clean out the bone marrow with cytotoxic drugs. So you're in an isolation facility for 2 to 3 weeks. But it works. And there's several advantages of autologous gene therapy. But the bottom line point is it works. I refer you to this paper in the New England Journal. But I can show this slide, which I show a lot to non-scientists. Because I said you don't need to be an immunologist to see that these children have no T or B cells at the beginning, and they recover, all but one of them recovered nicely after a few months, certainly enough to survive. But here's a baby I played with. You can see the family history. The family in Argentina had a boy who died of SCID with a routine immunization. That's the first sign of the disease. You die. Sorry. The second child was a boy but had the good X chromosome. The third child had SCID. It's this little boy. He was brought to Boston Children's, and here he is 5 years later. He's now 7 years out of it. The kid's fine. Okay? You can call it a miracle, but it works. And I see this now for other genetic diseases as well. But let me leave you with this. We can cure beta thalassemia, which is another, it's a hemoglobinopathy. We can cure sickle cell disease by gene therapy. I've actually there are 2 patients at Boston Children's that have been treated they've been treated and are successful for sickle cell disease. It's a lentiviral vector that expresses only in red cells an antisense RNA that knocks down a protein called BCL11A only in red cells. BCL11A is a repressor of fetal hemoglobin, and as a result, the children continue to make fetal hemoglobin and not the sickle hemoglobin. And I've met the first treated patient, a young man. He's now 22. He had a stroke when he was 4 years old because of the sickle cell disease. And he was in Children's Hospital almost once a month for his entire life, getting blood transfusions. He's now cured. Now, we're not done. There are millions of these patients around the world, and we don't know how to treat them all. We can't do it by bone marrow transplants. So many of you have seen a recent announcement. The Gates Foundation and NIH are pooling resources to develop gene therapies for sickle cell disease that could be administered anywhere in the world. I was part of that effort, and hopefully somebody will stand here in 15 or 20 years and even sooner and tell you how that works. So I just want to end, there are huge challenges here. This is the Chinese Organization for Rare Disorders that I privileged to serve on their board. You can see Kevin Wong, this gentleman here. He has himself a rare disease. And the whole reason this NGO survives in China is to try to convince the government to support research and even existing treatments on rare diseases. I just want to leave you with this very sad picture. At this conference 2 years ago, Brian Casper gave a spectacular talk on gene therapy for spinal muscular atrophy. It was done by Avexis. The company was recently purchased by Novartis for $8 billion. It works. Why am I saying it's sad? because these ladies here all have children in China with spinal muscular atrophy. They saw children in the United States literally climbing out of their cribs after treatment with with gene therapy. But this treatment is not offered in China. No gene therapy treatment is offered in China. And their babies will die. So I hate to end on a sad note, but I think it's important that students particularly, or your students, hear this, that rare diseases, understanding them and curing them is a huge unmet medical need. And also, it's a huge opportunity for biotech companies. Because as Genzyme and many others have shown, you can build a company and treat patients at the same time. So thank you, and again, I'm more than happy to take questions. Please.
Are there any major side effects to using the gene therapy in the treatment of sickle cell anemia?
Yes. I'm sorry, what's the question?
My question is the side effect for using...
Oh, the side effects. I'm sorry, I didn't hear you. Right now, there are none. Again, only 2 patients have been treated. And in the biotech class I teach, Dr. Ezrick, who runs the clinical trial, spoke, and she specifically answered that question from our students. But it's too early. And one would not expect a side effect, assuming the molecular biology is understood. In other words, the promoter is well characterized. The side effect that could be is you turn off BCL11A and other hematopoietic cells. But the promoter is well characterized. It's an enhancer and promoter for the beta globin gene. And what they're making is a small hairpin RNA that will target only BCL11A. So it's pretty good, but as with gene therapy in general, you have to watch the kids. The real danger is leukemia. The real danger is insertional mutagenesis of these vectors upstream of an oncogene. And you may know the history where in 2000, retroviruses were used to treat SCID. And they cured all 9 patients in France, but 4 of them came down with leukemia and 1 died. And that had to do with the E3 enhancer. The retroviral LTRs, the promoter enhancers, have a strong enhancer which will turn on a neighboring oncogene. And in all the modern versions of the virus, that enhancer is deleted. There's a huge advance in retrovirus vectors that I could go through, which eliminates that. So far, with all the lentiviral gene therapies, there has been no indication of leukemias. So all you can do is watch and hope. So far, so good. But it's not just sickle cell, it's SCID and it's thalassemia and the others. They're all using a similar lentiviral vector that has that offending enhancer deleted.
The children of a cured person will still have sickle cell.
They will still have in their genomes, their sperm or egg, the mutations, correct.
Okay. And so you would have to, this could work for a long time, you'd have to keep on using that kind of therapy.
No, I mean, no, the child, the child herself or himself that gets the gene therapy should be cured for the rest of the life. The offspring, depending on, I mean, these are autosomal recessive. So hopefully, if this person has a child, it will not be with someone who also has the mutation. I mean, this is where prenatal screening becomes critical in any of these genetic diseases. And that can be done. And virtually all the ones we're talking about are autosomal recessive, so it will work. Please.
I've talked to people who invest in rare genetic diseases, and one of their arguments is that the treatment is going to be very, very expensive, but that insurance companies will cover it because they won't have that many patients on their list with that disorder. But you end up with a really big price tag.
Yeah. Paying, okay, the answer is yes. I'll tell you how I think about it, which is really, I teach a course in the spring at MIT with a professor of finance. We teach it at the Sloan School. And he makes the analogy to buying a house. If you buy a house for $850,000, you don't pay $850,000 cash. You get a 30-year mortgage, and you pay off $400 a month. Now, why did he pick $850,000? That's the cost of fixing 2 eyes for the Spark blindness therapy. Each eye is $4.25. Okay? So the notion would be that the insurance company pays a certain amount up front, your down payment. I'm making it up, $100,000. And then you get a 30-year mortgage that you pay as long as the therapy's working. That's sort of where it's going. But the point is the treatment for many of these diseases over the child's lifetime, and we're talking about children's, is excessive, far exceeding the cost of gene therapy. And furthermore, the cost will come down. Wants me to move. Okay. So, but again, we haven't done, we're not done because only a handful have been approved. I see, yeah, go ahead.
Yeah, so I'd like to go back to your new startup, Rubius.
Yeah.
Not a startup anymore, but I was curious, what was the technology that was patentable?
Ah, what was patentable?
Because to people...
Modified, a whole variety of patents on modified red cells. First of all, we patented the culture procedure.
But theoretically, somebody could find another culture procedure.
It depends how you write the patent. But it's a culture procedure in which red cells have an antibody on their surface. That's a broad class. The way you write patents is you have broad classes, any red cell with an antibody on its surface, and then you list all the ones that you can think of or that you've made. So they haven't yet been litigated. They've been approved. And the claims are there. Ultimately, if someone tries working it around, it goes to court.
People have tried to engineer red blood cells before. I don't think you're the first.
It worked. No, we were the first ones to get it really, really working. In terms of conflicts of interest, I have served as an expert witness now in 7 very high-profile patent litigation cases. So I'm very familiar with, in fact, the last case, I invalidated a patent on the witness stand. So it can be done. And yet, a lot of it is trade secrets as well. You don't publish everything you do. So let's not go there. I see a hand in the back, yes.
Yeah, so referring to your last slide, so how do you foresee getting, that was gene therapy, to, at a global stage? Do you foresee, cooperating with the federal government, foreign government, just like how you cooperate with state government?
How do I foresee it? I do it. Next week I'm going to Hong Kong and giving several lectures there in Shenzhen precisely on this topic. I'm going back to Hong Kong and then Singapore in December. I'm meeting with political leaders. I do this all the time. I'm going to go to Taipei in March and give a course on gene therapies and try to convince them to do it. It depends on the organization of the country and how well receptive they are. You get into some interesting political situations. For instance, I spent a lot of time in China, and they'd like to believe that the Chinese are a single people, but they're not. There's been invasions from everywhere, and genetics in the Han Chinese are complicated. Genetics in the Indian populations are complicated. Probably more complicated than the United States. So you need to really think through what it is you're going to be doing. You need to get government support. Many countries have lots of indigenous isolated tribes that have their own genetic disorders, working with the governments in Vietnam for many years to work on some of their ethnic minorities, where again there's large fractions of consanguineous marriages. So a lot of it is awareness and the lack of either human geneticists or cultural anthropologists or everybody else. So a lot of what we do in China is just awareness, meeting as we can with governors and people like that. Yeah, a question.
I have a question for the HIV treatment. So as I know, so currently the treatment is the scientists use medicine to block the CD4 receptor in order to control the HIV disease. So I wonder, after you block the CD4 receptor, does the CD4 cell itself still working? Like for
what disease are you talking about? You mean
HIV.
HIV is CCR5.
CCR.
Yeah, HIV, the gene therapy, you can't knock out CD4. Gene therapy, HIV requires CCR5 as a co-receptor. And that emerged from gay men who were infected with HIV but never came down with the disease. And when they were analyzed, their bodies, they were mutant in CCR5, but they were perfectly normal. And then it emerged that you could delete that protein and block HIV infection. And the reason that is likely to work is that these individuals lack CCR5 and seem to be okay. So there's a whole group trying to develop this type of gene therapy.
But there's no way, like for now, there's no way to exactly cure the HIV disease. They have to take the medicine.
Again, I'm not an expert, but it's kept well under control by these combination drugs. And in fact, part of the argument is, if it's so well under control with these drugs, do you need to worry about gene therapy? And you see, I have the same issue with my grandson. He gets it's very similar. He gets the transfusions every 2 weeks. It's an inconvenience, costs his insurance company $300,000 a year, but he's fine. Would I subject him to gene therapy? Or when, in a year when he's 18, will he make the decision? Right now the answer is no. It's too risky. You see, even if it were effective, I'd still argue stem cell transplants. I might do it for HIV because it's a lethal disease. I'm not sure I would do it for Gaucher, which is not a lethal disease. You see the trade-off. Great, yeah, please.
So I don't know about the ethics of this. We were talking about gene therapy on patients with the disease. Do you know of any efforts to do any kind of germ cell therapies, or is that ethically or financially not?
Well, you saw what happened with that idiot in China. There's no reason to do it, which is the other point. I mean, leaving aside ethical issues of germline gene therapy, there's no rational scientific reason why you would need to do it. There are other ways of treating the disease, and we don't know the consequences yet. So you know, to me, it's always been cut and dry. The answer is no. You just don't do it. And then figure out why you don't want to do it later. But there's just no scientific reason why you would want to do germline gene therapy at this point. That's not true. I could think of a few weird cases, but let's not go there. Okay. Are we done?
I think so.
Okay. Thank you.